Good afternoon. It's my great pleasure today to introduce the invited speaker, Richard Flavell. I'm going to just say a few words of introduction. So Dr. Flavell is a Sterling Professor of Immunology at Yale and an investigator of the Howard Hughes Medical Institute. He first was trained in UK, where he obtained his PhD in biochemistry, left for Amsterdam for his postdoctoral fellowship first, and then in Zurich in the laboratory of Dr. Weissman. Among his many discoveries, and there are actually quite many, he discovered and was a co-discoverer of introns in cellular genes and showed that DNA methylation can control gene expression. Dr. Flavell was also the first to develop reverse genetics while still a postdoc in the laboratory of Dr. Weissman, an approach that he continued to develop quite creatively during his career. And this is actually what he utilized a lot to prove the function of genes in vivo. After his postdoctoral fellowship, he went back to the UK, where he was actually an investigator at Mill Hill, then going back and at one point to the States, where he decided now to join industry and was the scientific chief officer of Biogen for a few years. He went back to the academia a few years later and became the chair and the chairman of the Professor of Immunology at Yale, where he is now today. During his career, Dr. Flavell accumulated many seminal discoveries. And this actually ranged in the entire range of immunology, from inflammation, innate immunity, adaptive immunity, regulation of genes. Notably, Dr. Flavell identified key transcription factors that regulate gene expression of lymphocytes, and he also was able to discover the role of several receptors in innate immune responses, including TLR and NODs. More recently, his group actually has decided to be involved in the exploration of the crosstalk of the microbiota with the immune system and was able to show some important association between the microbiota, inflammation, and metabolic syndromes. In particular, he actually showed that actually dysregulation of the gut microbiota composition can lead to metabolic syndrome, including type 2 diabetes, for instance. He published over 1,000 papers, and this is likely not to stop. He actually trained a very high number of investigators that became leaders in the field. He received many awards. Actually, I cannot really list them right now. It's too many. He is actually an elected member of the Institute of Medicine, elected fellow of the Royal Society, elected member of the National Academy of Science, and so forth. He's really a giant in the field of immunology and really one of the most impressive and significant scientists of his time. And for all these reasons, it's a great pleasure for me to welcome him for this WALS lecture. Please help me welcome him.
That's the takeoff sign. So thanks very much. It's an honor and a pleasure to be here. This is kind of the epicenter of science, at least of biomedical science in the country and therefore the world. So, of course, it's a great feeling to be here and speaking to you all. I decided I'm going to talk today on work we really started around, I guess it's maybe 6 years ago or so. And it relates to sensing of microorganisms within us and the mechanisms that we use to do that, and then what happens when those mechanisms fail. So one of the great discoveries in the last, 20 years or so in the area of the immune system is the understanding of how microorganisms are sensed by the immune system. And of course, if you're going to be infected with something, before you can address the infection by dealing with it, you've obviously got to sense that the infection exists. And for those reasons, several people, and notably Charlie Janeway, really conceived that there had to be molecular mechanisms that would be able to do that, and he was viewing this in the context of adaptive immunity. And so in '89, he actually predicted this whole thing, and then by the mid-'90s, several labs, including Schulhoffman and Charlie's lab with Ruslan Mejitov, a fantastic young postdoc, and Bruce Beutler working with the LPR gene in mice, together discovered the toll-like receptors, starting ranging in expression from Drosophila to higher organisms. And really this opened the floodgates to the study of basically the recognition of microorganisms by multicellular organisms, ourselves included. So we became ourselves interested in the mid-90s in caspases. Caspases are, of course, enzymes which cleave a whole variety of interesting biological molecules, and we got interested in apoptosis and the role of caspases in this process, and in caspase 1, which plays a similar role but in inflammation. And this led us naturally over a couple of years to get interested in the family which is actually on the slide, which is the NLR, or nod-like receptor family. Now, nod-like receptor family is another cluster of genes which are involved in sensing of microorganisms. At least that's one of their major functions. They are found in the cytoplasm of the cell. Like one other family of sensing molecules, that's the nucleic acid sensors, which are molecules that can recognize either RNA or DNA. The NLRs actually are very good at recognizing perturbation to the cytoplasmic environment, either caused by damage to the cell membrane, in which case ionic composition changes, or a variety of other damages involving, for example, injection of microbial products into the cytoplasm, which is things like are delivered by salmonella or pathogens, gram-negative pathogens, for example, which use secretion systems. And study of the genes of this family by lots of people has shown that mammals have something like 20, 30 genes. We have something like 20. Mice have around 30, for example. But in early deuterostomes, there are actually hundreds and hundreds of these genes. And plants have similar genes, which are so-called R factors. And the functions, of course, in the things like sea urchins and sea anemones are not well worked out, but it's, of course, believed that they may have similar roles. In other words, at least partially involved in pathogen recognition, which is what, to a great degree, happens in plants. Now, the classification of these molecules is kind of explained on this slide, which is that the molecules in the top, that's pretty fuzzy. Is that too fuzzy for you, or can you see it? It's okay. This is what's called the NLRC group, and they have the prototypes, really, of the family, which is NOD1 and NOD2, very important because NOD2 is mutated, actually, in many people that have Crohn's disease. And these are molecules which have common properties with the other family. They have leucine-rich repeats, which are thought to play a role in ligand recognition. They have ATP binding domains, thought to mediate conformational change. And then they have oligomerization domains, which in the case of this C group is a card domain. The C, in fact, stands for card. So NLR with a card domain. And this is, as I said, this involves things like NOD1 and 2, and then some of these other ones, like this one, NLRC4, which recognizes gram-negative pathogens. The second group, which is a bigger cluster of genes, is called the NLRP group. They're called P because they contain a pyrine domain. It's a related fold to that of card domains, and it has a related function, which is to oligomerize this protein. So how does that work? Well, we have Jörg Chopp, a magnificent Swiss biochemist, to thank for the basic understanding of how this works, and it is caused by the juxtaposition of this NLR. This just happens to be one of them. This is NLRP3, the third member of this gene family in mice and humans. And this, what you see here is that upon activation of this domain, it associates with a pyrine domain of an adapter molecule known as ASC. That's this molecule here, which you can see has both kinds of oligomerization domain. It then can associate with caspase 1. Caspase 1 is one of several caspases which carry the card domain. Remember, card stands for caspase recruitment domain. Once this association occurs, this enzyme auto-activates, and then it cleaves its substrates to protease. And the substrates are the precursors of interleukin-1 and 18, and that leads to the production of active cytokines and an inflammation, which is downstream of that. And then a variety of other outcomes. Cell death being what's now known as pyroptosis is another outcome. This experiment just explains that, I'm not going to go into it, that this pathway requires these molecules in order to do this. This is done with knockout mice by Fayasutawala in our lab years ago. So if you fast forward a few years, we know quite a lot about what these molecules recognize, and the most striking thing really is if you look at the slide, you can see that numerous pathogenic organisms are able to activate this system going just through one of these NLRs. This is actually describing NLR3 in the main. And actually it's products of bacterial infection, products of viral infection, products of fungal infection, all will activate this system. But not only microbial components, also products of self-damage. So for example, necrosis activates this pathway directly. ATP coming from damaged mitochondria in necrotic cells will activate the pathway, and a variety of other products of necrosis, uric acid, a variety of different and more exotic things will do this. So in other words, it's a sensing system that can sense damage either provoked by an infectious or a non-infectious source. Because of this ability to recognize so many things and to integrate all these different inputs into very simple outputs, a couple of cytokines, dead cells, that we thought, and this goes back to the 2009 story, that the most scary place, actually, if you think about microbial recognition, that the scariest place in the body, of course, is the microbiota. These are the bacteria that live amongst us. Because at all of our surfaces, you all know this, there are microorganisms, and of course, in the gut, there are 10 to the 14 or something, these organisms. And so if you think about that, then you would imagine that there would be a necessity to maintain a homeostatic relationship with those organisms and ourselves. And so for that reason, we pose this question really, which is whether inflammasomes, that's what these things are called, inflammasomes, modulate the intestinal microbiota. And so two postdocs, Eran Elinav, who had just arrived from Israel, and Till Strovig, who had just arrived from Rockefeller, working in human immune systems at the time, got together in the lab and decided to address this question. I'm just going to summarize that because it's done a very long time ago. And this was the conclusion, basically. They were able to show this was indeed the case, and by mapping knockout mice against a property, which I'll just go into in a couple of minutes, they were able to show that mutants in these genes that I list here, so this is an upstream NLR, but it's a new one which hadn't been studied at that time, NLRP6, so that's, I showed you P3, this is an entirely different gene, so it's not another variant of that other gene product. This one is expressed selectively in epithelial cells, which I'll explain in a second. The adapter molecule I showed you, ASC or ASC, caspase-1, the target of activation of these NLRs, and a cytokine, interleukin-18, which is the target of cleavage by caspase-1. And when this pathway was dysfunctional, the microflora in the gut became dysregulated, and I'll show you what that means. Now, I'm not going to go into the experimental evidence for that because it's all published ages ago, but the bottom line is that in normal mice, now what you're seeing here is a depiction of the crypts in the intestine. This is the gut epithelium, and this would be the lamina propria here. And under normal circumstances, there are actually very few bacteria in the epithelial environment. And what we showed is that this pathway actually maintains that state of relative sterility. It's of course not pure sterility, but relative sterility of the environment. When this pathway is deficient, then this environment becomes invaded by these microorganisms, and that leads to the activation of inflammation. That's not surprising, of course, because once there are microbes, they will activate a variety of pathways. They will themselves cause damage. That will lead to the production of chemokines, and CCL5 was an important chemokine, which then draws in inflammatory cells. That leads to pathology. Inflammation drives pathology, damage to the gut, and that led to basically chronic inflammatory bowel disease in these mice, which was the assay that we used to measure. Another striking feature of this was that this phenotype, which was the acquisition of these microorganisms in this environment, actually was transmissible to co-housed wild-type mice. And that is because the number of these organisms increases enormously, something we can talk about later, for example, if necessary. But this dysbiosis, this change in composition that you can see here, by increasing all of these organisms, led to this transmission, and actually wild-type mice that are co-housed with these mice, well, this is this pair really on the picture, basically have the same invasion that the mutants have. Now, were these bacteria just a random collection of everything that's in the gut? That turned out not to be the case. So we went looking for what microbes were associated with this phenotype, this colitogenic phenotype, or phenotype that causes colitis. And what we found was there were very selective organisms, a handful only, which were enormously enhanced in numbers at the gut surface and in the feces, just in the gut, comprising percents of the total microbial composition. Mainly members of the family Privitellaceae, specific Privitella species, and a second group of organisms known as TM7. This is just a simple kind of principal component analysis showing that what I had mentioned to you earlier, that wild-type mice, when they're housed on their own, don't really have many of these organisms. But if you either analyze these mutants, these are mutated in that pathway, or wild-type mice which are housed in the same environment, then they get these very different properties with lots and lots of Prevotella and TM7. So that took us back to looking for these microbes, and as I said, just like that, the slide I just showed you, they are actually right there in the gut epithelium, and what you can see here is a transmission EM of a normal mouse. This is a crypt in the gut. These are goblet cells which make mucus, and if you look at that in a mutant mouse, you have a very different picture. This is a segment of a crypt. These are all bacteria. These black pigmented bacteria are very likely to be Prevotella. They're, of course, under EM, so you can't tell what they are without further analysis. But you can see they're very homogeneous, so likely there's an invasion followed by replication. And secondly, you have a hyperplasia of the cryptal environment, and in particular goblet cells. These are all goblet cells. Goblet cells are the cells that make mucus. So that was a depiction, really, by EM of what I'm showing you here as a model. Now, the mechanism whereby this worked, at least in part, we worked out more recently, and a wonderful graduate student, Marta Wlodarska, who was actually a student of Brett Findlay up in British Columbia in Vancouver, she came down to our lab to collaborate with Iran, and then also Ronnie Nowarski, who joined the lab in the meantime, and what they were studying really was what is the mechanism whereby this pathway prevents this invasion. And it turned out to be a very simple thing, actually, which is that the pathway is involved in the creation of the barrier which normally keeps the bugs out. So this is the epithelial barrier. This is the goblet cell, and these are actually the secretory vesicles which secrete the mucus layer, which keeps out most microbiota and most organisms in general, pathogens being the exception, because they, of course, have an ability to get through this system. But this consists mostly of a protein, MUC2, which can form this mucus-like layer, which basically protects the epithelium and keeps it mostly sterile. This is the barrier here between this inner and outer mucus layer, and this is where most microbiota are, separated by quite a distance from the epithelium. And the mutant mice actually had this very striking deficiency, which is that this mucus is stained green on this slide. I'm only going to show one slide just for time. But this is the inner mucus layer, which you can see here, stained green. These are goblet cells, and these are the secretory vesicles which bring which go to the surface, the apical surface of the goblet cell, and then release the material, the mucus here. But what we found, if you look on the NLRP6 knockouts, or all those other knockouts, they all have this same phenotype, really, which is they have vesicles which can produce mucus, but they are blocked in releasing this from the goblet cell surface. And so this mucin layer, this mucus layer, is in fact deficient in these animals. And of course, this immediately that makes it a bit more clear why this environment can be so easily invaded by these bacteria. But note, it's not all bacteria that invade. It's these quote pathobiont-like bacteria which are able to get into that environment. And so this was published about a year ago, so I'm not going to go into any more detail. But the story is really that we think that this pathway is controlling the function of this cell, and that the amount of mucus which is being secreted is actually regulated by an NLRP6 pathway, which in a sense is regulating and dosing the production of mucus by, we believe, by microbial recognition. In other words, if you see microbial recognition, it's going to tell you that you need more mucus to be produced. Now, we haven't proved that. That's just a sort of tautological argument, and it'll be very interesting to show whether that's indeed how it's regulated in the future. Now, dysbiosis, the alteration in the composition of the microbes in these environments, has been associated by a lot of people's work in numerous numerous chronic diseases in humans. And you can see a list of them on the slide. And many of these are systemic diseases, things like metabolic syndrome, diabetes, type 2 diabetes, obesity, inflammation-induced cancer. All of these outcomes have been associated with dysbiosis in the gut. And so it occurred to us, of course, that the dysbiosis that we saw in these animals that lack that barrier could have consequences not only on IBD, but also on these systemic diseases. So Jorge Henao Mejia, a wonderful postdoc from Colombia and Iran, El Inav, worked together to study that. And they studied first fatty liver disease. I'm not going to go into the details again. I think you all know fatty liver disease is a manifestation of metabolic syndrome. It's a big problem. It's something like 30% of the population has this condition, and most of people with obesity have steatosis, which can progress to liver damage, steatohepatitis, which can progress further to cirrhosis and hepatocellular carcinoma. So this is a big problem. And since the liver is the first port of call of the circulatory system from the gut, because that's where the food is going for detoxification and so on, then we reasoned that maybe this dysbiosis would influence this pathway. And so Jorge and Iran worked on this with various models, and I'll just show the answer, really, and this was their paper published a couple of years back now. And they again showed that all these mutant mice phenocopied this liver disease, this fatty liver disease. They were all susceptible to fatty liver disease. And again, it was an infectious disease. They could transmit the susceptibility to fatty liver disease to a harmless wild-type mouse just by putting it in the same cage. The bacteria from these mutants, which were elevated, would transmit to the wild-type and make that mouse susceptible, and phenotypically identical, actually. And the way that worked, which is in the story published, is as follows: that the dysbiosis in the gut led to leakage of microbial products like LPS into the liver, where it activates toll-like receptors, and this then led to the production of TNF-alpha, and it's the TNF-alpha actually turned out to be the driver for the fatty liver disease, and we showed that by mutating each of these genes. So all of the genes in dysbiosis, TLR4 or TLR9, MIDI88, and finally TNF-alpha knockouts. So none of the knockouts in this pathway get fatty liver disease, even though they're dysbiotic. And that's because the LPS, of course, cannot activate anything in that liver. So, again, it's just pointed out that this whole pathway, this is just from a news article, this dysbiosis that we find in these animals is enormously penetrant. And not only did it drive fatty liver disease, it drove type 2 diabetes, it drove obesity. Type 2 diabetes, work being done by Chung Jin, who was an equal first author with these guys. She showed for type 2 diabetes. And in work, I won't show it all actually, Bo Hu showed that, another great grad student, showed that even colon cancer driven by inflammation was transmissibly enhanced by dysbiosis. That was caused actually by the induction of interleukin-6 by the dysbiotic bacteria activating the inflammatory system, and the IL-6 acted directly on the epithelium, because when you took away the IL-6 receptor on the epithelium, you took away the enhancement. So all this leads to a picture that in these animals, and that we believe therefore in people, that a combination of all of these susceptibility factors, such as the diet, your microbial complement, and of course the genotype of the individual, can contribute to either a healthy situation or dysbiotic microbiota, which are a real problem. The host genotype, of course, is very important, because in the case I showed you, I showed you the most extreme phenotype for this purpose you maybe could have, because it lacks the barrier, which is essential. But in patients with Crohn's disease, they lack NOD2, for example, and NOD2 has important antimicrobial functions, and likely would contribute in a similar way. So this combination of these things, genetics, microorganisms, and environment here, drives this dysbiosis, which causes inflammation. And the inflammation could be read out as all of these features. It causes IBD, obesity, type 2 diabetes, cancer, and so on. And in the mouse system, at least, it's all transmissible. It's all infectious. And so all of this stuff is actually driven by these organisms caused by this combination of factors. So now, getting back to the microbiota in a little bit more detail, the organisms are vast, of course. This is... anyone want an email? Okay. And the question is, of course, what we'd really like to do in humans is to be able to understand what we were just describing for mice. And, of course, one can use 16S ribosomal RNA sequencing to get a census for what kind of bacteria are present in people. And a lot of that work's been extremely interesting and told us a lot about what organisms are present in various people. And that has told us that each individual has their own unique microbiota, again, as a consequence of their genetics, their microbial exposure again, their diet, drugs, like particularly antibiotics, of course. And it's pretty unique, the consortium that is in each of these individuals. And of course, our microbial self, this is the organism that lives with us, it changes and evolves much more rapidly than our own genome. Diet, for example, will cause it to change within a few days, in fact. But we basically have been evolving with these organisms for a very long time as a concept. And we normally, as I said, we have this symbiotic relationship, the homeostatic relationship. But what I want to focus on now is in people, the dysbiosis-driven phenomena, and how we can get inroads into how this process works. now, I just showed you that in the case of the normal mouse, we believe that we have this symbiotic situation where the microbiota has these basically these major components which are commensals. They are, in a sense, inactive in the sense they're not interacting with our immune systems. They're playing all kinds of important roles nutritionally and in many other ways, I'm sure. Then you have symbionts, which are mice, which are playing a role. Sorry, which are bacteria, which are playing a role in assisting, for example, in making regulatory T cells and stuff like that. And the organisms I focused on up to now, which are these pathobionts, which are become very common in disease, and then, as you see here, the ladder tips, and these drive disease. Now, the hypothesis, of course, is that this is just as important in humans as it is in those mice, yet we don't know much about what these pathobionts are. So the concept of good bugs and bad bugs is what we wanted to address in people, and to take our knowledge of this from the mouse studies. So individual members of the mouse microbiota clearly drive disease. I just showed you one example, is the Prevotella, which will drive colitis. Several people have shown that Helicobacter species, particularly in an IL-10 deficient environment, can drive colitis. And Dan Lippman's lab and many other groups now have shown that SFB, the segmented filamentous bacteria, can drive colitis and arthritis. These are three examples of pathobionts which are disease-driving members in the mouse. But, as I said, what we really want to know is what are they in humans? Based upon what we've been describing and based upon work of others looking at these issues, it's likely that there are organisms driving IBD, autoimmunity, type 2 diabetes, and of course it would be extremely useful to know what they were, because then you could get rid of them. So that was the goal of what I'm going to talk about now, which is really to develop some novel ways that we could identify disease modulating members of the intestinal microbiota. So the hypothesis we propose is that the host immune response to the microbiota can guide the identification of these disease-driving members of the microbiota. And I'll explain to you what we mean by that. Now, under normal circumstances, if you consider the situation of symbionts or commensals, these are clearly in homeostasis with ourselves, and there's not much going on. But when you, as you know, when you get infected by a pathogen, Salmonella, Shigella, all of these things, they invade the gut, they drive acute inflammation, and that leads to pathogen clearance, of course. That's the whole point of the immune response and the inflammatory response which occurs. But it seems reasonable that the organisms that we were describing in these earlier studies, here we go again, I don't agree to any terms or any conditions. Yeah. Should do the same thing. This should drive chronic inflammation, and they should drive inflammatory disease, because we're seeing this outcome. And so the immune system actually can distinguish between these good guys and these bad guys, and therefore disease-driving members of the microbiota are actually the ones that are driving the chronic immune response. So how can we use this property to identify these organisms? Now, one of the things that you would expect from these disease-driving members of the microbiota is, since they invade the epithelium and drive chronic inflammation, this itself should lead to adaptive immunity, because it's at the epithelial environment, specifically things like Peyer's patches, in which immunoglobulin A, which is the most important antibody in the gut, and the most antibody you make to anything actually, is produced. And of course, this would be an outcome. This is designed to protect against these organisms, whereas this inflammation, which is of course designed to protect, is what drives the symptoms. So IgA, as you all know, is a critical component of the intestinal immune response. Normally you have induction through sampling by dendritic cells, which pick up the microbial antigens. T cells will then drive a B cell response, and IgA will be produced, and it will be fed back into the lumen by transcytosis. As I said, you make a vast amount, we make a vast amount. It's 2 to 5 grams a day we are shedding in feces, so it's just a huge amount of material. And, but the point is, if you think about the IgA, what must happen is when it's sent out into the lumen, it will bind to the organisms which have driven its production. Because the idea is that pathogens invade, they're picked up by the DCs, and then you get a response, T-cell response, a follicular response, and then you get plasma cells which make IgA. And once that IgA gets out, it'll bind to these organisms. And you can see in gray, the IgA should bind to those organisms. And we can, of course, use that property. Did I go backwards? To do the following. Let's see now what we're doing here. Yes. So we decided that we should be able to use this property to take this property that the IgA would be bound to the bugs and to purify microorganisms that actually have IgA bound to them. That's the prediction. And the way to do that, of course, is just to take an anti-IgA, which is labeled with some kind of fluorophore, and take feces from an individual, stain them with anti-IgA, and then sort. And so we did a cell sorting experiment, and you can see here, this is from an actual experiment, that you get cells, these are bacteria now, which are coated with IgA. These are highly coated, these are less coated, and so on. These are uncoated. So most of the bugs actually are not coated very much, and several are highly coated. Now the question is that coating selective? In other words, is every organism coated equally, or are things like pathogens or pathobionts selectively coated? And that's what we of course hypothesized, that, and as you can see here, that the pathobionts would be highly coated, but these normal commensals would actually not be highly coated. And so, again, this leads to that idea for the hypothesis that you have disease-driving members of the microbiota, they activate inflammation, they activate IgA, but you can use them to identify who they are, and then that's of course the first step to doing something about them. And so the experimental setup was to do the following. We take the total fecal bacteria, we run them through the FACS, stained with that anti-IgA, and then separate them into two pools, IgA positive or IgA negative. You can see this is that pool, the IgA positive pool or the IgA negative pool here, and then just sequence the bacteria, and that will tell us the taxa which are actually coated with IgA. Now, the way we decided to express this is as a relative number. In other words, the IgA coating index, or ICI score. And that's the relative abundance of a given taxa in the IgA fraction, so in this fraction, divided by its relative abundance in the uncoated fraction. So if the bugs are highly coated, then this number will be high, and that number will be low, and therefore the ratio will be high. And if you take, for example, a thing like SFB in mice, then what you come up with is something like a number between 30 and 50. So pretty much all of them are actually coated. So, well, we had to prove that this was working, so we did a proof of principle first in the mice. And what we did is we took our standard model, basically. Remember, this is the experiments I was showing you a little bit back here, that you have dysbiosis in the mutants, and the wild types are normally okay, but that when you co-house the wild types, they become dysbiotic. And so what we did is to do an experiment where we took isogenic wild-type mice, took their feces, and compared the IgA coating of these taxa under this dysbiotic circumstance to the wild type here. And this is kind of the raw data from just looking at the total bacteria. So this is just counting just bacteria by sequencing, no IgA coding or anything. And you can see that there are these organisms like Prevotellacea I mentioned to you earlier. This is just a phenotypic. This is a DSS colitis experiment. So this is the wild type. This is the wild type co-house, so it's much more susceptible to colitis. And then we, of course, took the IgA positive and IgA negative fraction. So this is the wild type dysbiotic mouse, co-house mouse, took its total fecal bacteria, separated into these two fractions and sequenced. And then this is what you get. So you actually, this is showing the ratio of coating. This is all of these taxa. This is the total in feces, right? So this is everything that we sequence in feces. These are the IgA positives looking at frequency, and this is the IgA negatives. And this is the ratio. So if you look at the ratio, you get a very, very simple pattern. You get this one is not coated at all. That's a negative control. And then you have Helicobacter here, SFB. So it's the three class of organisms that we knew were associated with dysbiosis and dysbiosis-driven disease. Prevotellacea, which is the one I showed you earlier, Helicobacter, one associated with IL-10 mediated problems, and SFB, the one associated with all this stuff that Dan and other people showed. So the experiment actually worked beautifully and suggested that the method could work very well. And so the bottom line is that this coding index really does seem to predict the kinds of bacteria which we believe drive disease in the mice. So if that's the case, that really set us up to do something in people. So first of all, the conclusions here. So the IgA-seq identifies IgA-coded bugs. It's not at all random. It's highly selective. There's a very limited number of taxa which are highly coded. This is, by the way, T-dependent IgA. I didn't show you that, but if you have a beta-gamma knockout mouse, then you don't get these high affinity antibodies. All you actually get is low affinity anti-IgAs, IgAs, which are generally against things like lactobacillus, but much, much lower coating index. And then these colitogenic bacteria are the ones which are coated. It's not just a random selection there. So IgA coding really does mark the disease-driving members of the bugs, at least in the mice, and set us up really to do this experiment with people, which is to see if we take a variety of these diseases, do individuals with these diseases have organisms in there which are the same class of highly coated organisms, and can we show in some way that they play a causal role in those diseases? So that's the question. Can IgA coding be used to identify disease-driving members in people? Now, we decided to focus on IBD because it's the environment itself, and we have two very important diseases, Crohn's disease and ulcerative colitis, which have their own genetics. Lots of excellent genetics work has been done to show what susceptibilities are involved in playing a role in setting up the propensity to get these diseases. And of course, diet, and these things can play a role, but we wanted to focus on the microbial contribution to this. And so we basically took fecal material provided by our clinical colleagues, I'll mention a little bit later, from either healthy people, Crohn's patients, or UC patients. And in the initial study, we did a small cohort, really 21 normals, 27 Crohn's patients, and 8 UC patients. And then each of those fecal samples were subjected to IgA-seq. And this is what you get if you just look at the total sequencing. You get the usual quite complicated patterns. This is healthy, this is Crohn's and UC. You can see differences already. For example, some complexity down here. But it's a complex pattern. Each of these lines, by the way, is an individual person. Okay, so, but this pattern simplifies incredibly if you just look at IgA coding in these individuals. And so you find you can see that obviously there's differences between healthy people and these people with Crohn's or UC. And I'll focus on a couple of aspects of that. So first of all, microorganisms that are highly coated in normals tend to cluster. They are bacteria in general, which have been associated with a, quote, healthy state, defined loosely. They are present in lots of healthy people. They are less common, actually, in people with Crohn's and UC. But the other more striking thing was, in fact, that there were plenty of organisms present in Crohn's and UC patients which are not really present or present at only low coating levels in normal individuals. So, and this is the coating score. Remember, yellow, it means you're coated somehow between 30-fold to 50-fold. And then look at it, pick it in just a given individual, just to give you a sense of how many coated organisms there are. So this one here, for example, there's only 1, 2, 3 highly coated bacteria. So in general, it's a very simple situation with regard to the coating. It's just a handful of bugs compared to the number that are totally present in the intestine. So a much, much simpler outcome. Now, of course, that doesn't prove anything. It just says that there are bacteria which are coated with IgA in these individuals. But it set up the hypothesis, which is that IBD-associated bacteria, which are identified based on this coating property, do they play a causal role in driving colitis? And that was the focus, therefore, of our interest. Work from labs like Jeff Gordon's lab had shown that you can, in fact, take bacteria from people, and as long as you put them in germ-free mice, you can actually transmit a variety of properties. And a very visible paper back in 2006 showed that if you took bugs from lean people and put them into germ-free mice, you got lean mice, whereas if you took bugs from obese people and stuck those into germ-free mice, you could get obese mice as well, suggesting that the populations of these bugs contained organisms which could drive these properties, and that you could study human bacteria in mice. So we decided that this was the way to do this experiment, focusing on our own approach, of course, and just modified it to take the fecal bacteria and to take the bacteria which were identified as being IgA coated or IgA uncoated, put those into germ-free mice, and then to see, do you now get colitis? Because remember, we're taking these bacteria from somebody with Crohn's disease or ulcerative colitis. So does this property, this phenotype of the person, transmit to the mouse? Well, this became technically possible because Andy Goodman, who's at the Microbial Diversity Center at Yale, and Andy trained with Jeff Gordon several years ago. He's really a wizard at culturing these anaerobes, and he's worked a beautiful high throughput way of culturing large numbers of anaerobes from human feces in microtiter environments, and he can basically grow pretty much all phyla, most families, and most genuses, and most species within genus, at least 60% of the species within a given genus. So it's not everything by any means, but a lot of bacteria he's able to grow. Again, obviously there is room for improvement, but this meant that working with Andy, we could actually culture a lot of these organisms that were IgA coated or not in people, and then determine whether it was actually playing a role. So this is what we did. As I said, we characterized these organisms by IgA-seq and identified their sequences. And then, of course, we didn't take the organisms which have been sorted to do the infection experiment because they're probably dead because of the oxygen, et cetera, but instead what we did is to use the sequence information from this to match microorganisms which we grew. We grew around 100 to 200 colonies per patient. And I just realized I haven't even mentioned the names of the people who did this one. That all of this IgA-seq work was done and pioneered by Noah Palm and a collaboration between Noah Palm and Marcel de Souter, two incredible postdoctoral fellows. And so what Noah and Marcel did is to get 100 to 200 colonies per patient, sequence them, and then we could, of course, match those with these and then pick the right ones. So that gave us a clonally arrayed, taxonomic defined personal gut microbial culture collection from each individual person. And we knew the levels of IgA coating in the patient from which they were derived. So that's the question. We now take these bugs and put them into the mice, and do they cause colitis or do they maintain this healthy state? So what we did is to make a consortium of either uncoated bacteria or coated bacteria, because germ-free studies are extremely laborious. Each of these would have to be done in an individual isolator, and so that would be 20 isolators with God knows how many mice per sample, so that could be 100 isolators, so that would be turned into an impossible experiment. So instead we made consortia. This is 10 bugs and 10 bugs roughly, and made two groups. We took them all from patients. These are the uncoated bacteria, and you can see this is the coating level, so they're black, that they're uncoated. They're right down here, you see. These are the ones we actually selected. These all, of course, all grew. And from the coated ones, we took these samples, and these are the ones we actually selected. You can see, so this is B. fragilis, for example. This is a Streptococcus species, et cetera, et cetera, et cetera. And then we mixed them together in a consortium. So that gave us two consortia, the IgA positive and negative consortia. And then we colonized the germ-free mice. This worked pretty well. These are individual mice from the first experiment. So this is 5 individual mice, all housed in these isolators. And you can see that this is the inoculum and gives you the frequency of the bacteria as it was made. There's redistribution. One of them didn't grow in that particular case, but the rest did. They slightly redistribute, but the pattern is pretty homogeneous, and that recapitulates really work that Jeff and other people have done, which is that the mouse, you'll get a grouping which is kind of reflective of the mouse. And the same happened for the IgA positives. Most of these grew, and you got pretty homogeneous representation. Everything is represented to more or less the same degree. And these, again, are their names. And then when the first thing we asked, of course, is, well, do the IgA positive bacteria get coated with IgA again? In other words, does the mouse environment really reflect what happens in the human? And to a degree, it seems to do that. So if you wait until the IgA is, of course, forms, which is a couple of weeks, and you stain for IgA, you can see that the percent of IgA positive organisms is higher in from the bacteria which were IgA coated in the first place than it is with the IgA negatives. But there is some coating here, presumably because there is no competition whatsoever. They're the only bugs in town, so to speak. But the most important point is that actually the IgA positive bacteria did drive disease. And so what we did is to take those mice, as I showed on the slide, expose them to dextran sulfate to drive DSS colitis, and then compared the IgA negatives to the IgA positives. And the outcome was really striking. Here you can see this is histology score. So the IgA negatives basically don't drive anything much in the way of colitis. That most of them are completely normal. The histology looks pretty normal. But the situation with the coated bacteria is completely different. They give off-scale histology. The person, Li Ming, who actually read these, said she'd never seen as severe colitis in any of the any of the mouse studies that she'd ever read, and she reads a lot of these things. So these were highly colitogenic organisms, and of course derived from those patients. And here you can see some of the pathology scores. We also did the colon length, several other measures, all of them giving the same conclusion. So why do these bacteria, why are they colitogenic? What properties do they have? And this took us really back to the original studies I started off with, which is the issue of the NLRP6 mice and the fact that those bacteria invade the gut. And so that's what we predicted, that these bacteria should be invasive, and that's why they're colitogenic. So we looked actually in the guts of these mice. And for this purpose, what we use is a fish analysis, so fluorescence in situ hybridization, using probes for bacteria, but now a probe which will light up all of the bacteria. And what you can see, so here's the gut, here's that mucus layer I described earlier. And so in the IgA negative consortium, they are being kept out by this mucus layer, basically. You're not getting, all that red is the bacteria, this is the nuclei of the epithelium. But with the IgA positive consortium, these bacteria are invasive. They're getting into and through the mucus layer, suggesting that this property probably is an important one for driving the inflammatory response, which is actually what's causing the susceptibility to DSS. So that's the model we end up with, that basically, if you go looking at an IBD patient, they presumably are reflecting the same situation, that those bugs, the ones we found to be coated, are in fact invading the colon, driving IgA, and this is why these mice recapitulate that phenotype, and these get sick just as those patients get sick. So, just concluding from this, the IgA positive bacteria from the IBD patients will confer susceptibility to colitis. So therefore, they represent putative disease drivers in this disease. And that suggests that this the functionally defined subset of the microbiota can transmit actually a human phenotype to a mouse, enabling this to be done for multiple phenotypes. And therefore, that if we could work out ways to selectively eliminate these bacteria, they may ameliorate the disease and have therapeutic or, of course, or diagnostic value. There's one last thing I want to address, which is why do the bacteria which are assigned to the same species, which we see sometimes, some of them are coated differently in different patients? So B. fragilis was one example, I'll show you. And secondly, what about these IgA positive bacteria from healthy humans? What are they all about? So, as I said, when we looked at these 11 patients, this is B. fragilis, there were multiple patients that had B. fragilis, and these are highly coated, but these ones are not. This is mid-coating, this is uncoated at all. And so there's variability in here. So the question was, what was the driver for this variability? Is it the host? Is it bacterial genetics? Are they just different strains of bacteria, some of which are invasive, others which are not? Or is it the environment of the host, diet, what the person was eating, and so on? So we sequenced the genomes of a couple of these isolates. This is one, for example, from one patient. It's essentially uncoated, whereas the other one is coated heavily. It has a coating score of something like 37, which is almost as good as SFB. And this shows that they're different strains right away. You can see that these are, this is comparing each strain to itself and also to a type culture collection strain. And you can see that while they're called B. fragilis, there's a lot of genetic differences. Look at these clusters here, all through the genome. So clearly they're different strains, and maybe that's the property that is important. And so actually what we did is to take these individual strains of B. fragilis and to put them into, as monocolonized now, germ-free mice, put the uncoated one or the heavily coated one into the mice and see whether they indeed are healthy versus colitic when they get DSS. And the answer is that's exactly what you get, both by looking at the length of the colon, which I skipped on the slide, or the histology, which you can see pretty well. This is pretty normal colonic histology, but this is disrupted and basically inflamed colon. So these are just two strains. And this is a colitogenic B. fragilis, and this is an uncolitogenic one. And then finally, what about these healthy bacteria? What are they? They're IgA coated as well. Are they just problematic bacteria which aren't a problem in the people because the people don't have the genetic susceptibility, or are they qualitatively different? In other words, are they healthy? So we, again, made a consortium of healthy bacteria from normal individuals and put these into the mice. And here you can see this, and this is another experiment where we can combine, these are individual mice in individual isolators. So these are the IgA negatives. This is measuring colon length, which is the longer the colon, the healthier they are, basically. So these are healthy. This is the dysbiotic IgA-coated bacteria. That's the positive control. They are sick and have a shortened colon. But the IgA bugs from the healthy people actually are perfectly healthy. Those mice are not getting sick at all. So actually, what we postulate that these bacteria might well be is some kind of symbiotic organism that may drive some actually protective state. So we're now investigating that in some more detail. And so, as I said, what we're thinking now is that in IBD you get this invasion by these microorganisms, these dysbiotic organisms. They drive these Th1, Th17 responses, which cause disease. But you have, in a healthy person, you have a predominance of those healthy bacteria. Again, this all has to be worked out in detail. And maybe their job is to do things like elicit the production of Tregs by mechanisms that many people have worked out. Yasmin and colleagues all over the world working out the ways that these things work. And that in a healthy individual, this predominates over these other organisms and contributes to this homeostatic state. Whereas in the IBD patient, because of the genotype, these guys are basically winning out. So then just to finish up, this immunological profiling that we're able to do exploits the ability of the immune system to tell good from bad, and that this functional profiling of these microbiota can reveal previously hidden patterns that really weren't visible by just looking at the whole microbial constellation. And it really supports the concept that gnotobiotic mice colonized with defined consortia from humans can really be used to establish causal connections between microbiota and human disease. I should emphasize that at the moment, I think, while I think the evidence, I'm quite encouraged that the evidence is suggesting that these organisms really are involved in disease in people. Of course, it doesn't tell us at this stage whether they initiate disease in this genetic environment, or whether once disease is established, that they're making it worse by for example, finding an environment that's inflamed and where that makes things better for them. That's not possible to distinguish from this. But I think the fact that they do this in mice really suggests that they are causally involved. And we've started to analyze these bacteria as individuals, and that some of these are really nasty, very, very bad bacteria, at least for mice. And then hopefully this can be used to validate and study the effects of these disease-modulating members of the human microbiota in lots of circumstances beyond these diseases. So to finish up, these are really the heroes of the latter story. I mentioned while I was going through it that Iran Ilinav and Till Strovig got the whole project going. They were joined by Jorge almost right in the beginning, and all that earlier work was done by them. And this whole IgA story really is these are the heroes. Noah, who really got the whole thing driven. He drove it himself. Marcel de Soute, who joined right in the beginning. So they were working together. They cooked this stuff up in the animal room. This collaboration. And I mentioned Andy Goodman. Andy is a wizard. Without him, we'd have been absolutely nowhere. And these are the people with him. Ashley Shade actually helped Noah. He met Ashley walking his dog in the park, and she turned out to be an informatics whiz, and so she helped Noah and Marcel to do the analysis and taught them at least to do the analysis. And then, of course, we'd again been nowhere without human samples, and we've had a very long-standing collaboration with the wonderful Judy Cho, one of the great people in this field, and we're still continuing to work together. And Inga Peter at Mount Sinai, and Jian Zhong, Elizabeth Ruggiero, and Wei Zhang at Yale, all were involved in getting good human samples. And then finally, Li Ming, who did all that histology, was essential, of course, to the interpretation of all this. So thanks very much. I'll be happy to take questions.
So I've been told to remind people there will be actually a reception after if you want to discuss Dr. Flavell, but now the floor is open for questions.
So fascinating story, as always. And I have a couple questions related to the mucus. So one is, does the NLRP6 effect that you described on production of mucus by goblet cells, does that involve any of the cytokines that are known to affect mucus production, IL-22 and so forth, or is it independent of that?
Yeah, good question. It seems to be cytokine independent, and I should have said, I can't remember if I said it, it's IL-18 independent. So this is the pathway down to caspase. It acts through autophagy, and the NLRP6 pathway is controlling autophagy, which we believe is regulating the release of the vesicles, but we don't know how that happens. Autophagy mutants have the identical phenotype to the NLRP6 knockout. They have a thin mucus layer. And actually another group showed independently in a paper in EMBO journal that mucin, the mucus layer is defective in different autophagy mutants. So that's the connection, although there's a lot more mechanistic work needs to be done. But it seems to be cytokine independent, I think.
Okay. So then the connection to the second part of your story is, it seems as though the ability to penetrate that inner mucus layer is key. And that could be either at the host level in terms of the quality or quantity of the mucus, or at the bacterial level in terms of the ability to invade.
Yeah.
So do you know if there are differences among the different strains of B. fragilis that you looked at, for example, in their ability to invade? And in patients with different diseases, differences in the mucus that's produced.
Right. So I think that point is absolutely correct. It has to be both, right? Because it's even with the compromised mucus layer, it's a very selective numbers of organisms, a handful only. So there have to be properties of those organisms which are confirmed that. From the sequence of the B. frag at the moment, we haven't really learned very much, so we don't know. But of course, that's the direction. That's where we have to go, is to identify what are the properties encoded by these bacteria which are causing disease. They may there's still quite I should have said the obvious, which is there's not one bug, right, for Crohn's. There's not one bug for UC. You can see that. But there may be common properties of these organisms. So, there's a ton to do still, but we haven't done it. Yeah.
So, Richard, if I understand correctly, in different IBD patients, the bad bugs may be different.
Yes.
So, do you foresee some sort of a personalized medicine whereby the IgA coding would identify each person's bad bugs, which then could be specifically targeted for extinction in one way or another?
Yeah. So we haven't done enough patients to know yet. So we're in the middle of doing 400 Crohn's and UC patients. We should have them done by the end of this week, actually. And that's when we're going to know how complex the situation is. Some of the organisms actually turn out to be pretty common. And when we've done more, we've analyzed around 75, and the new ones I haven't seen yet. But in that larger group, there are some organisms which are present with as high a frequency as 50%, especially in UC, for example. And so it could be moderately straightforward, or it could be, as you say, have to be personalized. And in which case, you would imagine the strategies, of course, would have to be worked out, but you would certainly involve diagnosis of the organism followed by whatever you do, right? So there would be a kind of a two-step procedure. So we're a long way from declaring victory here on the aircraft carrier, so to speak. Yeah.
So, Richard, can you tell me something about population dynamics? If you mix the IgA tagged bacteria, pathogenic ones from humans, with either IgA positive or unassociated from healthy humans, can you suppress pathogenicity?
Yeah. So we've only done one mixing experiment yet, and we mixed the IgA negative bugs from Crohn's with the IgA positive. So the first two consortia, and they still develop disease, although it is somewhat attenuated by the presence of the negatives, presumably some competition.
No.
The really interesting experiment is the second one, of course. We haven't done it yet, which is to take those healthy bugs and see if they are suppressive.
That's the issue.
Yeah, yeah, that would be very interesting. We haven't done it yet.
Raising the doses.
Yeah, exactly. And it needs to be quantitative, of course, but of course, dose response curves with a germ-free mouse is not the easiest thing. Well, it's easy, but it's expensive.
I'm going to take one more question, and then the rest of the discussion can happen during the reception.
Richard, I'm glad you mentioned at the end that these bugs could be secondary
Of course. Yeah.
to the disease. I was wondering if you did the following experiment, namely, take patients who are very thoroughly treated and have no existing disease anymore due to anti-TNF therapy, whatever, whatever therapy, and then see if their functionally bad bugs go away.
Yeah, that's a very good question. Certainly haven't data yet. We have a very good cohort of patients that are being longitudinally studied. Not every one of them, but and of the 400 samples that we have, some of them are certainly longitudinal samples from the patients. I don't remember if any of them are, quote, symptom-free. I would fear that they might not be, because they're not the ones that show up at the clinic so much, right? But that's an experiment that could be done, and that would be great to know. Yeah.
Well, thank you very much, everybody, for